Good afternoon, everyone. I want to welcome you to the latest in Axiom Healthcare Strategies Town Hall Series post-ASCO. Today, we're going to be talking about non-small cell lung cancer. We're looking forward to an active discussion and really appreciate you joining us. You'll see a feature on Zoom where you can ask questions. Please feel free to submit your questions that way, or you can also submit your questions directly to Axiom by emailing me at rs@axiomhcs.com.
Great, thank you, Robin. Welcome, everyone. And we are very privileged to have today Dr. Roy Hirth from EL Cancer Center, as well as Dr. Suresh Ramalingam from Winship Cancer Center at Emory University. As Robin had mentioned, please send your questions through the Q&A feature on your Zoom at the bottom of your screen. You can also email questions to rs@axiomhcs.com.
Thank you, Hafez. I think I'll take it from here. It's really great to be here, and welcome, Suresh. How are you?
Very good, Roy. Good to be here as well.
So now that we've introduced, we're going to go through an agenda that includes immuno-oncology trials, and who better than you to help us discuss 227 and 9LA, and then we'll go through the CITYSCAPE, then among others, then we'll talk about targeted trials. I've heard about the ADAURA trial. I can talk about that a bit. CAM5, the Destiny trial, and then really unmet needs, future rationale, combinations, new pathways, and investigation, and then hopefully a robust Q&A session. So let's start with the next slide. So the FDA has granted 7 approvals for metastatic lung cancer treatment thus far in 2020. What do you think, Suresh, of this? Pretty incredible, the speed that these are getting approved.
Mind-boggling, right? Just to think about the month of May. This is not even all of 2020. Just in the month of May, we had 7 new indications for lung cancer, and this includes targeted therapy and immunotherapy. So these are substantial steps forward in our ability to personalize treatment for patients and also have options in some settings. For example, in frontline non-small cell lung cancer for the high PD-L1 patients, now we have atezolizumab as another option on top of pembro, which has already been in use. The combination of nivo, ipi plus chemo is another option. And the exciting news is there are many more drugs that are down the road that hopefully will join the ranks of newly approved drugs for lung cancer.
No, it really is great, and to have drugs like cabmatinib and selpercatinib available, especially with COVID and clinical trials down, I think is very good. There was also a new approval, wasn't there, with pembrolizumab to give it every 6-week dosing. So that was interesting as well. Again, convenience is always an issue, but maybe more so during this current period. Okay, let's move on.
Yeah.
Let's move on. Oh, go ahead.
I was just going to say, like times of COVID, the Q6-week approval of pembrolizumab has been a godsend because it helps patients stay home longer and have less exposure to the healthcare setting, and for our staff to keep the physical distancing in the clinics. These sort of interventions are very helpful.
Right. I'm also intrigued by ramucirumab or erlotinib, the Relay trial. I cut my teeth on this about, God knows, the year 2000, about 20 years ago, when we look at erlotinib and bevacizumab together, and slightly different story, but same idea, and interesting to see that that's now available. So really more and more options for patients, and what we're going to talk about today is a little bit about the data and how to use some of these drugs. So what do you say we go to the next slide, see what we got? So practice impacting lung cancer. So CheckMate 227. So this is nivolumab and ipilimumab versus platinum doublet chemotherapy as a first-line therapy for advanced non-small cell lung cancer, and a 3-year update. So 3-year data. So I'm going to turn this to you, Suresh, because you actually presented this.
Yes, thank you, Roy. This is an update for this trial based on a median follow-up of close to 43 months. This trial is very familiar to folks. The initial results have been published in the New England Journal in 2019. Just to refresh everyone on the clinical trial design, this was for patients with newly diagnosed stage 4 non-small cell lung cancer. It included both adenocarcinoma and squamous cell carcinoma patients. These patients could not have an EGFR or ALK or a targetable driver mutation, and they had good performance status. This trial enrolled close to 2,000 patients, and the randomization was based on PD-L1 expression. The PD-L1 expression categorization was at 1% level, so those who had greater than or equal to 1% expression were randomized to chemo alone, nivo alone, or nivo plus ipi. For patients with PD-L1 expression less than 1%, the randomization was to chemo alone versus chemo plus nivolumab versus nivo plus ipi. The primary endpoints for this trial were two. One was PFS, specifically looking at high TMB patients, and that has been reported upon and published. What we're going to talk about is based on the ASCO presentation that focused on the overall survival for the PD-L1 greater than 1% population, which was another primary endpoint. The next slide shows us the 3-year update. This is comparing nivo-ipi versus chemo versus nivo in the PD-L1 1% or greater. Few points to make here. One is there was a statistically significant and clinically meaningful improvement in overall survival with nivo plus ipi. The long-term follow-up continued to show that the benefits that were seen in the previous report were maintained. There was hardly any change in the hazard ratio. What is new information also here is the 3-year survival rate. At 36 months, you can see that the survival rate with nivo and ipi was 33% compared to 22% with chemo. The nivo alone group also did fairly well at 29%, but if you can see, the hazard ratio is 0.9. So really the ipi-nivo group is the one associated with the best outcomes here with the 3-year survival rate of 33%. If we go to the next slide, we can see results for patients with PD-L1 expression less than 1%. Now, this was a secondary endpoint in the trial. You can see here again that the 3-year survival rate with nivo and ipi was 34% compared to 15% with chemo. So there's more than a 2-fold higher survival rate at 3 years. The hazard ratios for the low PD-L1 patients or negative patients was even better at 0.64. The median survival was improved by about 5 months when one compares this to chemotherapy. Next slide. So this slide summarizes what we reported at ASCO with a minimum of 3 years of follow-up and a median of 43 months of follow-up. The 3-year survival rate with nivo and ipi is 33%. And this is very similar between patients with PD-L1 expression negative versus those that were 1% or higher. We also noted that the duration of response was substantially longer. For instance, for patients treated with nivo and ipi, the median duration of response was 23 months for PD-L1 positive group, compared to only about 6.5 months with chemotherapy. We found that the safety profile was consistent with what had been reported previously. There were no additional safety signals. Most of the autoimmune adverse events were noted in the first 6 months of therapy. Once patients got beyond that, there was no major increase in autoimmune adverse events. And finally, the FDA just approved, as we saw from the previous section, the combination of nivo and ipi for frontline treatment of non-small cell lung cancer patients for the PD-L1 greater than 1% group. You can also say that based on CheckMate 227, the combination is effective for the low PD-L1 expression group as well, and is now available for treatment based on NCCN guidelines. So I'll stop there, Roy, and would be curious to hear about your thoughts. Where do you think this fits in the clinical paradigm of non-small cell lung cancer?
Absolutely, and congratulations on this presentation. 30% actuarial survival at 3 years in metastatic lung cancer. Wow. When I was growing up in this field, if you had any patients alive from a trial at that point, it was a miracle. So are these selected patients in some way, or is there something that we need to learn about these patients? Because it sounds like it would really help to know some molecular markers or some way a priori who should get these two drugs.
Absolutely. Looking for biomarkers to even narrow down the benefiting patient population continues to be a key theme. In this trial, we enroll patients as long as they were those with newly diagnosed, no prior chemo, and did not have a targetable driver mutation. They were enrolled, and it's a global trial, so it mirrored what we see in our practice. When you just look at the outcomes for the control group, which is the chemo-treated patients alone, that group did really well in this trial. So the comparisons are consistent with what one would expect to see for patients in their respective clinics.
Right. Why didn't you include a Nivo chemo arm, someone asks, in the PDL1 greater than 1% population?
Well, at the time this trial was designed, there was a lot of interest in frontline single agent therapy, Nivo alone. Remember, Pembro was studied in comparison with chemotherapy. CheckMate 026 was ongoing, which compared chemo to frontline nivolumab. And at that time, we did not have the CheckMate 026 results. So the trial at the time of design, it felt like having Nivo alone as a comparator to chemo was very relevant. Uh, Adding a fourth arm would have made this that much more complicated in the design of the trial and the statistical sample size, particularly from 2,000 patients, it would have jumped up by another several hundred patients. So at that time, the Nivo versus chemo comparison was still relevant. Now, in the PDL1 negative groups, we did have the chemo plus Nivo because Nivo alone for that group was not seen as a substantial upgrade over chemo based on knowledge at that time.
I see. So someone's asked, what's the biggest impact? I guess I'll start, from my perspective. I think this is a pretty nice result. Certainly, it looks good in both the PDL1 high and the low group. It's a shame that it doesn't have the approval there as well, but I gather that was because of multiple statistical comparisons and power to look at all the different arms. Is that the reason?
Absolutely. I think that would be a big factor.
But I see many patients who don't want to have chemo, even COVID notwithstanding, and this is a non-chemo option. And I think we have a trial here at Yale, 592, CheckMate 592, that actually is including biomarkers, sequencing, transcriptional analysis, CITYSCAPE, stool, a whole bunch of things to try to understand in the frontline setting who these patients are. But I think these are nice data, and this puts another option out there for us to discuss with our patients in the frontline setting. You agree?
I absolutely, and I do think that the option of giving this combination and preserving chemo for another line of therapy resonates with a lot of our patients that we've discussed these results with in the past few weeks.
Right,
and then I was also going to say that the durability of the responses with the Nivo-IP combination is something that sets this apart. When we look at the chemo plus PD-1 or PD-L1 trials, the durability of responses is in the order of 9-10 months. What you see here of 20 months plus also is an appealing reason to consider this for patients.
I agree. And there was something at ASCO, right? This looking at patients with COVID and lung cancer, they don't do too well if they've gotten chemotherapy, right? That was the TerraVolt data.
Yeah, the TerraVolt data and other studies have also showed that one of the risk factors for higher mortality, if a cancer patient were to develop COVID infection, was receiving chemotherapy within 14 days prior to the infection. So during the COVID pandemic era, that's highly relevant.
Right, and we saw some stuff at the AACR too. Okay, let's move on. We don't want to. So now we have CheckMate 9LA, nivolumab and ipilimumab, plus 2 cycles of platinum doublet chemotherapy versus 4 cycles of chemotherapy as first-line treatment for stage 4 recurrent non-small cell lung cancer. Martin Reck presented this right after you. So you're obviously in the virtual room there, Suresh. You want to just do a quick look at this? What does the LA stand for?
Boy, I don't know what LA stands for. I think the coding systems have become so complicated.
Maybe they designed it in California, but okay. Yeah, let's hear about the trial.
So this was looking at using 2 cycles of chemo with nivo and ipi in the frontline setting. This takes the approach of chemo plus immunotherapy combination, but with the nivo and ipi, the thought was you don't need to give chemo forever. We can just give it for 2 cycles and continue nivo ipi on forward. So this was a typical frontline study. This included patients with squamous and non-squamous histology. Patients with driver mutations were excluded. Comparison was to chemo versus 2 cycles of histology-appropriate chemotherapy with nivo and ipi. The dosing of nivo and ipi here slightly different, but very similar in terms of actually administered dose of the drugs. So nivo was given at 360 q 3 weeks. Ipi was given 1 milligram per kg every 6 weeks. And the treatment was continued for a maximum of 2 years in the immunotherapy group. The primary endpoint was overall survival. The secondary endpoints included PFS and objective response rate by independent assessment. The trial included patients regardless of PD-L1 status. Patients were stratified based on PD-L1 expression of less than 1% versus greater than 1%. At the time of this report, the median follow-up is approximately 12 months. There was an interim database lock that occurred way back in October 2019, at which time the median follow-up was shorter at approximately 8 months. Next slide. This is the overall survival Kaplan-Meier curve. You can see here that there was a statistically significant improvement in overall survival. The curves separate early and stay separated throughout. The median overall survival was 15.6 months with chemo plus nivo plus ipi compared to 10.9 months with chemo. The hazard ratio was quite robust at 0.66. Next slide. Here you see the results broken down based on the PD-L1 expression level. In the lower PD-L1 expression group defined as less than 1%, hazard ratio was 0.62, favoring chemo plus ipi-nivo. And you can see that in the PD-L1 greater than 1% group, the hazard ratio was 0.64. And another piece to keep in mind here is if you look at the PD-L1 greater than 50% group versus the 1 to 49% group, you see consistent benefit. Even in the 1 to 49% group, the hazard ratio was 0.61. So overall, this shows that every group, regardless of PD-L1 expression, derived comparable benefit. And what you don't see here is the histology-based look at hazard ratios. Both squamous and non-squamous patients had favorable outcomes with the chemo plus nivo-ipi combination. Next slide. So this is the summary slide that Dr. Rex showed. It met its primary endpoint of overall survival with a hazard ratio of 0.69, highly significant p-value. The results were not just statistically meaningful, but were also clinically meaningful. The magnitude of benefit was consistent regardless of PD-L1 expression. There were no new safety signals. The combination was generally tolerated well. And this also has led to a new FDA approval, chemo plus nivo plus ipi, with 2 cycles of chemo being the chemo component for frontline treatment of non-small cell lung cancer.
That was wonderful, and let's have a few minutes of discussion on this. So we have a couple of really great questions from the audience. They're really asking the right questions. The first is the toxicity of this quad, chemotherapy plus the ipi-nivo. How well tolerated is that, and how logistically complex is that, and is it doable? I guess the whole reason for only doing it over 2 cycles was to sort of minimize that, right, Suresh?
Yes, and the main difference between the 9LA and the CheckMate 227, even though we don't want to make cross trial comparisons, is that the survival curve for the nivo-ipi alone versus chemo plus nivo-ipi, if one recalls in the 227 trial, initially the survival curve dips a little bit and then it starts looking better than chemotherapy. Whereas with 9LA, they remain separated and consistently go in a favorable direction for chemo plus nivo-ipi. So the initial 2 cycles of chemotherapy is helpful in making sure that the patient is benefiting and it in some way augments how they respond to immunotherapy afterwards. We did not see any concerning safety signal. The treatment schedule is patient comes in once every 3 weeks, so that is not affected by giving 2 cycles of chemo. You do see the myelosuppression that one expects to see with chemo and the autoimmune adverse events that one expects to see with immunotherapy. But there was no synergism and toxicity between these two groups, groups of drugs. Overall, I think this is a tolerable regimen. And when I think about where I would use it in my practice, this has some parallels to what we do in the greater than 50% patient population. For most of my patients, for greater than 50% population, PD-L1, I would go with pembro monotherapy. But there are some patients who have significant disease burden, their symptom burden is very high, and I feel like their course is declining very rapidly, that we want to give them the benefit of an aggressive combination. So for those patients, I would give chemo plus pembro. And when I look at the 9LA regimen, that's exactly how I feel it could be used in the clinic. If the decision is to go with Nivo and IPI for a given patient, that should be okay. If your patient has bulky disease and is having a lot of symptoms, having the 9LA regimen there might be helpful.
Right. We have several questions like that. That's the $1 million question here. Where does it fit in? Certainly it's no surprise it was positive. If the ipi-nivo was positive versus chemotherapy, you would think this would be, unless there was a toxicity concern, which there wasn't. It's hard for me to see where this will be used because people are so much using chemo plus pembro. But remember, that's only for non-squames, so this would work for squames too. But I agree with you. There are sometimes people with high PD-L1 where you want a little bit more bang for the buck because you're worried that they might have early symptoms, and it is that the two parts of the curve. You say it very well. We've got to get over that hump to get to that flat part of the non-proportional hazards that we deal with IO. So it's good to have it out there. My feeling is let's have these things available and continue to do work to figure out who they're best in. More biomarker work, maybe real world data will help us with some of this. But let's move on. Let's see what we have next. CITYSCAPE. Okay, maybe I'll discuss this one, give you a little rest. So I'll turn it over to you. You can ask me some questions. Yeah.
Yeah, I mean, obviously, as one looks at what are the promising immunotherapy combinations, this study clearly comes up to mind. This was reported at ASCO 2020. So, Roy, walk us through what your thoughts were on this trial.
Well, we're all looking for new things. What's new, and TIGIT, is certainly pretty new. There are 7 or 8 groups now that have it, probably more, because that's all I know of. So I'm sure there's twice as much going on, 3 times. But let's go to the first slide. So this was a randomized study, and it was patients with frontline lung cancer, untreated, the EGFR ALK wild type. Basically, they don't have any abnormalities in EGFR or ALK. And PD-L1 was actually looked at by TPS, so it was using the 22C3 antibody greater than 1%, and which is a little bit different than the way atezolizumab has been used in the past. But actually, there are some data that were presented at ESMO that shows that the assays are relatively equivalent. And the patients are randomized to the atezolizumab with the TIGIT, it's tiragolumab, or to a placebo plus atezolizumab. And the primary endpoint, they're treated without crossover until progressive disease or loss of clinical benefit. Co-primary endpoints of overall response and PFS, which is what we have. Secondary endpoints of safety, duration of response, overall survival, patient-reported outcomes. So, and then of course, PD-L1 status being looked at. Next slide. So here are the data. So here you can see for the full trial, it's a phase 2, 135 patients. Response rate was higher in those patients who got the atezo plus the TIGIT, 37% versus 21% for the control, and it is statistically significant. And then when you broke it down by PD-L1 status, this was quite interesting. So for the PD-L1 50%, and I've reviewed the literature on this, and we'd love to have a TIGIT assay and know which patients have high TIGIT. But if you actually look, you can see that there are associations between the two running together, PD-L1 and TIGIT. So here's 66% response rate, which is quite good. especially when you have a control group of 24%. So this was quite compelling. But in the PD-L1 low group, 1 to 49%, doesn't look like there's much difference at all. It's a statistical dead heat. So unclear exactly why you wouldn't see the benefit in both groups. You have any thoughts on that, Suresh?
No, Roy. I think some of the things that I was thinking about in this trial is even in the high PD-L1 group, the control arm somehow didn't do quite as well as what one would have expected, a 24% response rate. So small study, we have to look at it directionally. I don't think it's powered enough to tease out differences between various subgroups.
Right, I've read that too, that atezolizumab underperformed. It's easy to underperform in a small trial, because the vagaries of the randomization of, we don't really measure PD-L1 that well. You've got to have PD-L1 for these drugs to work. So if it's PD-L1 0, it's just that we're not seeing it. So there are some issues in small trials and imbalance. But the bottom line is, there does seem to be quite a benefit. And I think on the next slide we have the PFS, which is even more important. It was funny, all you had going into ASCO was the median numbers, which, we had a median of 3.88 months and 5.5 months, and I saw that in the abstract, okay. But you see the hazard ratio is 0.56. There seem to be some tails on those curves, and that's the ITT on the left. That gets me pretty excited. And then if you look at the TIGIT plus atezo versus the placebo atezo in the 50%, the hazard's 0.3. So again, small trial, and of course in the PD-L1 1 to 49, there is no benefit statistically, as you would suspect, given that all the benefit was in the 50% group. But I think it's enough data for me to see it through to another trial. And I think what's the new trial called? The Skyscraper. So the CITYSCAPE needs a few more skyscrapers. Is that what's going on, Suresh?
That's my understanding. And Roy, one question I had was, we're looking at PD-L1 expression, which we all know predicts for benefit with immune checkpoint, specifically PD-1, PD-L1 blockade. Are there any specific markers that might be of interest for the TIGIT antibody?
Well, I think, being a checkpoint molecule, I think we should have an immunochemistry assay or RNA-seq or something like that. And I'd love to see that. And I think big question I have, I missed it. Maybe you saw it. They presented the phase 1 data at AACR this week. Over the last 3 days, we've had AACR, and I attended much of it, but couldn't still have some patients to see and other things to do. But I know that those phase 1 data are out there. I don't believe that there were many phase 1 single agent responses, but that would be important to know. Are there single agent responses, and can you enhance that with a biomarker?
Absolutely, and this is really one of many combinations that are being studied. It's great to see something promising that builds on immune checkpoint inhibition and will be hopefully be confirmed by follow-up studies, the Skyscape. Are there any other combinations, Roy, that you've seen that are exciting for this patient population?
Well, I think the combinations with VEGF inhibitors look good. Things like lenvatinib, the LEAP studies, certainly ramucirumab with pembrolizumab looked attractive. Love to see that go forward. Certainly in renal cancer, these combinations are active, but that's a VEGF-driven tumor. But I think the VEGF combinations are good. Cabozantinib, which gets VEGF, but a lot of other things too. I think all of those looks pretty good. Citravatinib. So agents that target the microenvironment, I think, would be high up on my list. But I'd love to see more trials with biomarker results.
Great. I think as we wrap up the immunotherapy section, Roy, I couldn't help asking you about where you see these drugs going in early stage disease. We have now a number of phase 3 trials, either completed or nearing completion in the adjuvant therapy. What are your thoughts on those trials, and when do you expect to see some results there?
Well, as I'll talk about in a moment when we do ADAURA, when I first started taking care of patients with lung cancer, 1994, 93, Tom Fry at the Dana-Farber at that time used to show me papers and writings that the best drugs always should be moved earlier in disease. That's where you'll have the biggest impact. And that was, of course, the model with chemotherapy. I think with biologic therapy, targeted therapy, we've seen that now with targeted agents. I think the same is going to be true for the immunotherapy, bringing them forward and using them earlier, with one big caveat, Suresh. You've got to both have earlier disease, the good drug, and the biomarker. And I'm not so sure we have the biomarker. So it worries me a little bit. I'm very excited about the Alchemist trial. I think the biggest contribution of that trial probably will be the immunotherapy arm that accrued so quickly, and kudos to you and your group for that. But the question is going to be, do you need a biomarker? Is it enough, just to take, PDO, does biomarker matter at that point? And I don't know. So that's going to be the big question. And does the risk outweigh the benefit? But I think it's definitely the way to move forward. I love the idea of neoadjuvant therapy. So that's a great setting, because there you can see what's happening in some of the neoadjuvant trials. And, I even have patients coming in, looking for neoadjuvant, therapy now, because, it's become that well known that the results of immunotherapy pre-surgery.
Great. Let's now move to the next part of our talk, our discussion, which would focus on targeted therapies. Next slide, please. Yeah, so Roy, number of targets in lung cancer, the last count is 7 targets with FDA-approved treatment options now, and potentially many more, KRAS, HER2 knocking the doors. What's your take on how much impact targeted therapies have made in lung cancer?
Oh, it's been huge since the first EGFR inhibitors went into clinic in 1996. Now with third generation drugs, like osimertinib, targeting the mutant receptor, it's just amazing. And I think the key thing is this only makes a difference if you look for it. And I would bet if you polled 100 centers, very few are testing for all of this. Certainly, if people send out to some of the foundation medicine platforms and others, they get that, but a lot of the in-house systems might not. So one push, and of course there's access to care, which is given the current crisis, we realize healthcare could be so much improved in our country still. We've got to make sure patients with lung cancer get these tests done, because all of these targets, or most of them, like RET, for example, phenomenal results. The problem is you have to be sure you know this in advance. The thing about targeted therapy, and I'll put that up there to be proven wrong, is that no one's really ever cured. You can treat people for long periods of time. They can do well. Certainly, we're looking for agents that target the brain. The brain is an area where we know that these drugs don't get up there at high enough levels in the CNS unbound that they can be active. But it's very intriguing. It's very promising for patients, but resistance will develop. But I think the benefits of targeted therapy are enormous, and certainly if you have a target, I would use a targeted agent before I used IO, and maybe someday Suresh will combine targeted therapies with the IO. Some early growing pains there, but that might be very useful.
Absolutely, I think you make some great points, Roy. One, make sure you're doing NGS, not just single gene testing, because there are so many targets now, and make sure that one waits for these results to come back before you start a patient on treatment. If you had to start somebody on treatment, just use chemo. Don't get into chemo IO, because if after cycle 1 you have to switch to targeted therapies, there could be some overlapping toxicity issues. And for a patient with targeted therapy is better than immunotherapy or chemotherapy.
Right,
and those
absolutely.
are major take-home messages for metastatic disease. Now let's shift gears, talk about what all of us think is the highlight of ASCO 2020, which is the ADAURA trial. You can call us biased because we like lung cancer information. Certainly there were other abstracts in the plenary session, but to me, this was the most important one. So Roy, tell us about the ADAURA trial.
Right, so ADAURA is osimertinib, third generation TKI EGFR in patients with stage 1B to 3A EGFR mutant positive non-small cell lung cancer after complete resection. So in the next slide. So here's the trial, and a couple of things, didn't expect to present the trial this year. The trial was not due for an efficacy analysis. It was having its yearly safety analysis, and the independent committee noticed an imbalance in terms of efficacy in favor of the study drug. And I've done a lot of these committees myself over the years, and very rarely do you stop a trial for this reason. You're usually stopping it because it's futile, but they felt compelled to look at the efficacy because they saw signs of such positivity. The trial was powered for a hazard ratio of 0.7, keep that in mind, a 30% improvement. And then they saw the data that I'm going to show you now and unblinded it at the level of the data. and that happened around April, early April, and then of course you've seen the data already, and more to come in the future. But basically, patients with stage 1 to 3A non-small cell lung cancer, you can see the age, exon 19 or L858R EGFR mutation. They have to be non-squamous, non-small cell lung cancer, which is where most of those mutations occur. Brain imaging, everyone had brain imaging, so we know about brain mets. That'll be an important sub-study analysis at some point. And you have to have interval between surgery and randomization was 10 weeks. But if you had adjuvant therapy, which some of the patients did, that could be 26 weeks. And then patients were stratified by the stage, the EGFR mutation status, and the race, Asia versus non-Asia. And then they received chemotherapy when appropriate. As you can imagine, most patients with stage 2 and 3 received chemotherapy. The stage 1's probably many fewer. And then they received osimertinib orally, 80 milligrams a day. There was standard dose, or placebo, with a planned duration of treatment 3 years. And the treatment was continued until disease recurrence, treatment was completed, or discontinuation. And you can see the follow-up, that's important, until recurrence, and then week 12 and 24, then every 24 weeks to 5 years. The primary endpoint was DFS by investigator assessment in the stage 2 and 3A group. Actually, there were about a third of the patients were stage 1, a third stage 2, a third stage 3A. So the primary endpoint was in the 2/3 of stage 2 and 3A disease. And as I said, powered for 0.7. Secondary endpoints, which will come, and we have some of those now, DFS at 2 years, I'll show you that, 3 years, 4 or 5 years, and then ultimately overall survival. So I already explained why it was unblinded. So on the next slide, here are the data. And, I expected this would be a positive trial. It's an active drug. It's got good CNS penetration, safe, tolerated, can be given for a long period of time. Dr. Ramalingam authored a wonderful paper in the metastatic setting that showed that it was superior to relatlimab, gefitinib. But wow, here the primary endpoint is 0.17 hazard ratio, so an 83% improvement in disease-free survival. It's mature at 33%, and most of those events are in the placebo arm, as you would imagine. So I would say look at the 2-year mark. I feel pretty good about that maturity. 90% versus 44%, so doubling at 2 years of those patients who recurred. Other lung, brain, liver, bone, all that's hoping to see that very soon. Really just a fantastic result, and the reason why it made a plenary at the ASCO virtual meeting. On the next slide. So here's the whole group. So even when you add in the stage 1 patients with their much better prognosis, you still have a hazard ratio of 0.21 and a 79% improvement, 89 versus 53% at 2 years. And this is, of course, a little less mature because you've added in the stage 1 patients, but still just a very compelling result. Patients living with their lung cancer without recurrence, which I think certainly means a great deal. Next slide. And here's the DFS by stage, and just you can look at the curves, or you can look at the table in the upper right. So hazard ratio is 0.5 and significant for stage 1, despite the good prognosis of stage 1. About 50% of stage 1s will recur. Stage 2, about 66%. Stage 3, about 75%. Stage 2, 0.17. Stage 3A, 0.12. They're really a very fantastic result. Next slide. So this is the first targeted agent in a global trial to show a statistically significant and clinically meaningful improvement in disease-free survival. 79% reduction, as I mentioned, in the primary endpoint. 2 years, 89 versus 53%. And this was seen whether patients received adjuvant chemotherapy or not. We didn't show that for the sake of time. But there's a nice forest plot in there where everything is positive in favor of the study drug. Didn't matter whether the patients received chemotherapy or not. Those data will be presented in an upcoming Congress. The safety profile was consistent with the established profile of osimertinib, which was quite good. So this is we believe this will provide a highly effective, practice-changing treatment for patients with stage 1, 2, 3 EGFR mutation lung cancer, which is a lot of patients. Worldwide trial, 30% of patients probably have early disease. Probably another 10 to 15% have EGFR mutants in the U.S., 20 to 30 in Asia. So really very exciting to have this at ASCO this year.
Absolutely. Congratulations, Roy. This is, I would say, one of the most stellar results that we've seen in lung cancer or any cancer for that matter in a long time, showing an impact of an intervention be that dramatic. Now we have the DFS results. When do you think the survival results will be available? That's the first question people ask us when we talk about ADAURA.
Well, absolutely, and I'm the first to, I showed a early survival curve, 5% maturity. Um, it's good that we have to wait for survival. It means people are living longer, and it's an early report of a trial, so it's going to be some time. The exact amount, I think, there'll need to be discussions with regulatory authorities regarding at what level the next look is taken. So that's all ongoing now, but I think it's going to be a while. I think in talking a year or two or something in that effect. But in the meantime, we'll continue to follow these patients closely. That's the key thing here now. The good news is the trial was unblinded at a point where the last patient had been on for more than a year. So the median duration of therapy is 22.3 months. So even if someone progresses now on a placebo, there's really no reason to start an EGFR inhibitor. And there will be some crossover though, Suresh, when patients progress on the control arm, or they will all end up getting the EGFR drug as they should. And the one thing that's really nice is AstraZeneca has made it clear that they will make sure every one of those patients gets that drug. But I think despite a crossover and having to wait, I think with DFS like that, I'm quite optimistic we'll see a survival benefit as well. But in the meantime, you have patients living without brain metastases, bone, liver, other lung. So really nice that we get the data out as early as possible so that physicians and patients can use these data to make decisions.
Absolutely. So you touched on this briefly. There's a question from our audience, Roy, about some information about placebo in the control group. Can you clarify? I know the patients received standard chemotherapy and then were randomized, right? So
right, I get asked that all the time.
Yeah.
I get asked that all the time. Well, chemotherapy is the standard here, and we're part of a lot of those studies. It's okay, but it's not great, right? Improvements. You need a calculator to show the absolute benefit to patients. These different nomograms, and it's maybe 5%, maybe 8-10% max improvement in outcome. There was no standard therapy with EGFR agents. If not, I would not have been comfortable doing this study. The RADIANT trial, published in JCO 7 years ago, maybe more, erlotinib versus placebo, no benefit. Hazard ratio PFS was 0.6, 0.7, no benefit. Granted, that was a subset analysis of that trial because they weren't selected for EGFR mutation. But up to the point, EGFR mutant patients were just basically told, we know you have the mutation. Sometimes we didn't even know that because people weren't even testing. Now I suspect, there'll be more testing at the time of surgery. Some patients would come to us, I'm sure they came to you, and they'd want to have the drug, and it wasn't standard of care. We did not do it. So placebo was very reasonable here because there was no standard of care. I don't think it will be a reasonable control arm anymore in future studies, obviously.
Absolutely. And patients who had received surgery and were candidates for adjuvant chemotherapy were randomized after they got chemotherapy. And also the fact that the study was done in parts of the world where chemotherapy is not particularly favored by patients. So that could explain why a good percentage of patients did not get chemo. But what I was struck by is the forest plot you mentioned, where regardless of whether you get chemo or not, osimertinib versus placebo comparison showed a very consistent hazard ratio.
Absolutely, and I think that's important, and we will need to look at some of the more nitty gritty, and a big trial like this, 800 patients and worldwide trial, will keep us busy for a while, looking at some of the different subsets, and that's all ongoing right now. You can imagine the database is probably, was so young. You've done many of these trials early at the time that we presented, but we're accumulating more data right now.
Another question, Roy. This is referring to the CTONG study that compared gefitinib to cisvinorelbine in China and in the adjuvant therapy setting, where there was a hazard ratio of approximately 0.6 for DFS, but at ASCO, Professor Wu presented the results and there was no survival difference. So one of our audience members wanted to get your comments on how does that apply to ADAURA?
Well, it's a very different study. First of all, it's gefitinib, not osimertinib, very different drugs. I believe osimertinib to be more active, probably to be more active killing tumor cells. Also, to be able to be given for longer periods of time. The drug was given for 3 years because it's better tolerated. That said, the other trial was asking a question: chemotherapy versus targeted therapy. And also, I suspect there are some more metastatic patients in that trial. I'm not sure that they did complete restaging before they did the randomization, which we did in the ADAURA trial. So we were pretty sure we're dealing with locally advanced disease. But it's on the table now. We're gonna have to figure out: do patients need chemotherapy? There are going to have to be other trials now to look at this, and I think there will be. What do you think?
Absolutely. I think so too. I think these results are rather dramatic, and the fact that osimertinib is best-in-class EGFR TKI makes this different from interpreting some of the other trials with earlier generation of TKIs. The bigger question, Roy, is EGFR is seen in 15% of lung cancer patients, so we can do trials like ADAURA. Now, as we talk about other targets that are seen in 1% and 2%, like ROS, MET, and RET, how are we going to do adjuvant trials, and whether we extrapolate from trials like ADAURA to use them in earlier stages of the disease?
So I guess we need the lung map early stage approach. we need the biggest, and Suresh and I work on lung map together. But the biggest funnel we can get. 1% of lung cancer, 1.5 million cases a year worldwide, 30% early disease. I'm a little too tired to do the math in my head now, but it's a lot of patients. So you could imagine that if everyone was getting sequenced and we knew that result, and we were efficient in putting people into trials, one could imagine we could do this type of thing. And we probably should, and get to a world where you get your sequencing done at the time of surgery. We know what the molecular abnormality is, and I have no reason to believe that this wouldn't work for an ALK or for a RET, but harder to find. The advantage here is, that this is 10-15% is low, but it's not horrible. But 1 to 2% is pretty hard.
I think clearly we need to encourage patients to participate in clinical trials, and we also need to have the right trials for patients to participate in. Let's move on to and collaborate across not just institutions, but across various parts of the world to do these kind of trials in smaller subsets of patients. Roy, let's switch gears, talk about the antibody drug conjugates, a group of drugs that have been around for a while. Nothing has really made it to the lung cancer landscape in an approved manner. We saw the results for the CEACAM5 targeted antibody drug conjugate. Can you walk us through those results and some key take-home messages?
Right, I'm not as familiar with these, so maybe we can do it together.
Sure.
Okay, but let's go to the videotape, as they used to say. Next slide. Actually, would you mind getting started on this?
Yeah, sure. Happy to do this. I mean, the antibody drug conjugates are basically monoclonal antibodies tagged with a toxin directed against a specific target. In this case, this product targets CEACAM5, which is expressed in lung cancers. So they did a dose escalation trial followed by a cohort expansion, specifically for patients whose tumors expressed CEACAM5 at a level of 2+ or more. Next slide, please. And they included all histologies. And what you see here are some very early and promising data. In patients whose tumors had high CEACAM expression, the objective response rate with monotherapy was about 20%. And in the moderate expressors, the response rate was somewhat lower at 7%. When you look at disease control rate, it's approximately 60% of the patients in both groups. Next slide shows us the swimmer plot for these patients. Once again, you can see that the median duration of response for the high expressors was approximately 5.5 months, and nearly 1 out of 5 patients were still on treatment. The median number of cycles were 8. When you look at similar data for the moderate expressors, the median duration of response had not been reached. A smaller group of patients numerically compared to the high expressors. But regardless, you see some favorable outcome for 30 to 40% of patients in both of these groups. Next slide. So this is an antibody drug conjugate, promising. I think it is going to be potentially applicable for a biomarker selected patient group based on expression of CCAM in greater than 50% of the tumor cells. And the tolerability has been good, and they are now moving forward to a phase 3 clinical trial in the salvage therapy setting.
Right. So this is exciting. You know, these antibody drug conjugates are sort of, you know, something where they often will have activity, right? Because you are targeting a, you know, molecule enhancing the tumor. The question is going to be the durability, I think. Do you agree?
Yes, absolutely. And what is the ability to give these drugs on a sustained basis? Because you do end up with some cumulative toxicities with the antibody drug conjugate because of the toxic load that you add to the antibody. But nevertheless, it's promising, and we wait to see definitive trials confirm these results and have some options for salvage therapy of non-small cell lung cancer.
How common is the CCAM5 overexpression?
I knew you were going to ask me that question. I believe it's about 20 to 25% of the patients, but I may not be fully accurate on that.
Right, but yeah, thanks for taking that one. But yeah, I think it's I like ADC. You know, they're always gonna, you know, it's basically delivering chemo. You know, a guided laser bomb, and the question is going to be durability, and then how viable is the target, you know, and how specific is it to the tumor.
Yeah.
Okay, let's move on. We have a few more minutes left, and what is your destiny, Suresh?
Well, this is, I thought, one of the more exciting studies after ADAURA in ASCO 2020.
I agree. I really like this one.
It showed activity for HER2 mutated patients. So, Roy, do you want to talk through these?
Yeah, this one I know pretty well. So next slide. So Destiny 1, I love the name, open label phase 2 trial, unresectable metastatic lung cancer, relapsed or refractory to standard treatment. Now here's HER2. So HER2 is 1 or 2%, but again, it's an important group of patients expressing or activating mutation, no prior HER2 targeted therapy except for pan-HER TKIs, because some of the TKIs actually get HER2 as well, and 2 cohorts. One was the HER2 expressing cohort, so IHC 2 or 3 plus, and the other exactly equal group of HER2 mutated patients, and the primary endpoint was response rate. And let's go to the data on the next slide. So here you can see the best change in tumor size. This is the HER2 mutated cohort. This looks pretty good here. You can see the results, and the waterfall is the waters are all flowing down, and then you look at the so really good waterfall plot. You know, a good number there. I think it was like 30 or 40%, right, of PRs, and then you look at yeah, then you look at the response rate over time. The arrows indicate patients ongoing, so good durability. So I told my group, find out who makes this drug. Let's get it in our clinic. Next slide. And then you can see, so here's the mutated cohort, the progression-free survival and the overall survival. And you can see the median PFS in the mutated cohort, 14 months. You can see the error bars there, but that's a pretty good number for refractory lung cancer, an amazing number, quite frankly. Normally you would see 3 or 4 months. And then the overall survival, not yet reached. So this is something I think we need to watch carefully.
Absolutely, Roy. When I saw this, I felt that this could be the 8th or the 9th target in non-small cell lung cancer with an effective option. When you look at a response rate of 60% plus and a median PFS of 14 months, even though in this trial, these patients had all progressed on prior chemotherapy, and majority of them had received 2 prior lines. So this is a very heavily pretreated group of patients. In our practice, if this drug was available, I would say these results merit use in the frontline setting for a HER2 mutated patient.
Couldn't agree more. Okay, let's see if we have anything else. If not, we'll have some discussion. I think we have discussion. So here are the conclusions. I think we already said that. Yeah, 61%. Sorry, I would have been happy with 40%. 61%. And you can see some of the numbers are not reached. Safety profile was good. Some GI and hematologic AEs were low grade. Drug-reduced ILD, that's a little bit of a something. Now this drug's been used in breast cancer, of course, and you know, it's approved there. But drug-related ILD, that's something we're pretty used to working with that. Do you think that this is going to be a big problem here, Suresh, or we know about ILD from other drugs we use?
Yeah, I do think that the results on this study are based on a relatively smaller group of patients. So as more patients are treated, we'll have greater clarity. The other thing is the dose that was used in this trial, 6.4 milligram per meter square, is slightly more than the 5.4 milligram dose that's used for breast cancer patients. So there's now ongoing efforts to understand what's the right dose of the drug for lung cancer patients. The majority of the ILDs were grade 1 or 2 in this trial. So clearly something that needs to be followed, and early recognition and management is going to be key. And with greater number of patients, we'll have a better quantitation of how common this is in lung cancer.
Okay, very good. So next slide. I think we're in questions now. So now we have discussion points. Let's see if we have anything after this.
I think we have the conclusion slide after this. And
Okay,
we have
so
answered
questions.
most of the questions that are coming up from our audience so far.
Okay, so you've been good about that. So let's see, we're at 1:55, or almost at the hour. Let's check the questions. I don't see too many that we haven't answered. So why don't I do the IO conclusion and you can do the personalized conclusion. So
sure.
With C227 and 9LA, we've seen that IO therapy is an option for patients. I think there's no doubt about that. So Suresh presented, and as someone not directly involved, I would say that clearly we're looking for ways to avoid chemotherapy. That's always been the goal. The fact that chemotherapy plus immunotherapy is the first combo out there, it's a little counterintuitive, but it does work. But we have some pretty good drugs out there to knock off the perch, but here we have something that looked quite active. There are ongoing studies across tumors to better understand how to treat patients who are not responding to immunotherapy. So mechanisms of resistance and possible combination approaches, absolutely. I would say for both points, personalization is going to be important. Who goes out? Some people can get a whiff of pembrolizumab and be alive 10 years later, and some won't even make it through one cycle, even with a 50% PD-L1. So we've got to figure out who those patients are. Suresh, targeted therapy?
Absolutely. So the two abstracts that I think will make us talking about for a long time and potentially enter the clinic and impact the ADAURA results, of course, very stellar improvement in disease-free survival up to 3 years of post randomization. I think these results will lead to a change in treatment practice. If osimertinib is approved in the adjuvant setting, I believe that it'll be used in that setting. Obviously, we want to see what the impact on survival is as the data become mature. And finally, for HER2 mutated patients, the ADC that we discussed, which is the TDXD drug, has very high response rates, which bodes well for this patient group. Again, early data, 42 patients, but encouraging nevertheless. So we hope to see more results from expanded groups of patients. So we will actually have an approved targeted option for HER2 mutated lung cancer, which is about 2 to 3% of non-small cell lung cancer, non-squamous patients. We have not seen any drug that's as active as this in the space today. So these are very exciting results in the personalized therapy front from ASCO 2020. And it opens up a lot of new possibilities for our patients, and also for what we have to look forward to in the upcoming months in 2021.
Absolutely. Well, I think it's an exciting time. It's good for patients. A lot of research going on. Trials will continue despite this horrible virus, and we will figure out what the best combos are. And the fact that we have so many new drugs, I think, is very exciting too.
Yep. All right, thank you so much. It's been my pleasure to have this conversation with you, and I hope our audience learned our take on ASCO 2020 and will join us at the next opportunity.
Absolutely. We'll bring the panel back again after ESMO to highlight some of the ESMO data. Any closing thoughts as we kind of learn to survive through the pandemic, to your clinician friends out there in the communities on how to address some of the lung cancer patients through this?
All I can say is do the best therapy you feel is needed for the lung cancer patient, and don't compromise. If you can avoid chemotherapy safely, do it. If you can put off visits a little bit less frequently, do it. But I would say, I'm sure Suresh will say the same thing at Yale. Our offices are safe. We're being cautious. We're distancing. Everyone's being checked, and the lung cancer is not going to go away. We've got to treat it. We've got to treat it aggressively. Suresh?
Absolutely, I agree with those completely, Roy. Cancer doesn't wait for COVID, so we have to do what we can to help our patients along during these difficult times.
Great, thank you for your time. And the YouTube video will be available in the next 24 hours, and we'll see you again after ESMO. Thanks, everyone.